Hi class. I just wanted to quickly go over the Gizmo because I know that it's only given you 5 minutes, and so you still have to fill out that Gizmo sheet, and maybe some of it you can do um, even without the Gizmo, and I'll try to figure out the subscription issue. But if you're struggling, here's a video. So what you're going to see me doing right now is putting together the DNA strand. Um, As you can see, the outside of the DNA strand is made up of a sugar. This is a ribose sugar and a phosphate. And then attached to the ribose sugar on the inside is the nitrogenous base. These are the letters A, T, C, and G. So I'm going to keep adding sugar, phosphate. Let me go back up. Sugar, phosphate, another sugar, and a phosphate. Has to go on the leading. Okay, there we go. So there is one half of my DNA strand. Now I'm going to match it complementary. When you need to take a picture, you use this little camera thing right here. A goes with T, so I'm going to put a T right here. And then I'm going to add a phosphate. And then G goes with C. Your order might be different than mine. That's okay. I just want you to understand what the structure is like and how it works to carry both information in its bases, like an alphabet, and also how it, sorry, I'm multitasking. To keep my office hours up, I forgot. And also how it can manage to copy itself. Okay, so you can see I'm making these pairs here to make my DNA strand. And then at the end of this, what would take place is it would twist like a helix. And that's based on the attractions between different parts of the molecule. Okay, so here's my DNA molecule. Each of these is called a nucleotide. And there's my last phosphate. Missing a phosphate right here. I have 2 minutes left. Let's see if I can get this done. So then you would take a picture. I'm going to add my DNA helicase. So release the enzyme. It made the two sides of the DNA split. This is for DNA replication. And then I'm going to release the enzyme for making copies of these two strands. And so you can see these are the two original strands. And then what I add is going to lead to the replication phase. So I'm going to start by pairing the sides. So T goes with A, G goes with C, C goes with G. Let's see if I can finish this in another minute and a half. I'm racing here. I know this will take you a little bit longer than 5 minutes because you haven't done it before. Okay. Almost there. C's and A. Okay, so now you can see I have two strands of DNA and they are identical to each other. This is how replication works. Half of the original strand, half of the new, due to the complementary base pairing. That's the A with the T and the C with the G. That's called complementary base pairing. Allows for DNA to both carry a message and to easily replicate itself. And this is also going to be important as we learn about transcription and translation this week. I hope this helps.